Hello, and thank you for the opportunity to speak today at today's event. My name is Raul Silva, and I'm a scientist at CN Bio Innovations. Today, I will be presenting on the topic of bridging the gap, how human MPS improve the translatability of NASH drug discovery. But before going into the details of our research, let me just provide some context in the details of our research. And some information about CN Bio. So CN Bio are a bioengineering company based in Cambridge, UK, and we specialize in single and multi-organ microphysiological systems, also known as MPS. Our technology, also known as organ-on-chip, was licensed from various research groups, including MIT and Vanderbilt University, with whom we still have a long-standing collaboration with. And the mission of our company is to be at the forefront of developing new medicines using predictive models that can provide human translatable data. But why is this work important? So, as many of you are aware, drug discovery is a challenging process, and high clinical failure rates due to safety and efficacy are usually common. So, in fact, there are approximately 90% of drugs and compounds that fail in clinical trials, with usually unsuspected safety issues and lack of efficacy being the major causes. So basically, currently, the current preclinical approaches include in vitro 2D cell cultures, as well as 3D spheroids. On the third one as well, we also have in vivo animal models, and each approach has its pros and cons. So in vitro models can be high throughput, cost-effective, and human relevant, because they use human cells. However, they lack the complexity of 3D tissue and blood perfusion. They also cannot be cultured for extended periods of time, and they provide limited data content. On the other hand, when we look at animal models, they do provide complex organ systems, but they also offer longer longevity and provide extensive amounts of data. However, they still lack the human relevance, as they are not human, and they are often obviously low throughputs and are very often expensive to have. So the high clinical failure rates of drugs resulting from the current preclinical models, it highlights the need for new approaches in drug discovery. So building a reliable and robust preclinical assay requires considering several factors, as we see here. So the assay should be more cost-effective and accessible than animal models, and it should also provide high throughput capabilities. It should also be physiological and human relevant, and being able to emit organ microenvironments and address population variability and immunological effects when using cells derived from patients. And finally, the assay should be able to show the effects of the medicines and compounds being tested, and therefore providing an efficient tool for drug discovery. So how do current MPS... So current MPSs come in different shapes and sizes, and they have a lot of different similar features. So these features include the use of human cells and tissues. So these are usually primary human hepatocytes, or primary human cells, including hepatocytes and other cells, or patient-derived tissues that are often grown in 3D and being in a 3D environment. They also have fluidic flow in order to replicate the blood flow and have a better microarchitecture of the organ that we want to try to replicate. This is all done by electrostimulation, either by fluidics, pumping of biomechanical stimuli and such. And finally, one very important feature is that we need to be able to control the therapeutic exposure. So basically, we need to be able to dose at different time points and being able to monitor the effect of these drugs, either with sensors or by sampling of the samples. So how do MPS compare to current preclinical methods? So when we compare MPS or organ-on-chip with the other three classical models that I've spoken about, we see that organ on chip have better human relevance, as we not only uses human cells in 3D. It also allows for the microenvironment with the fluidics. Also shows like better complexity and longevity compared to other models. Higher throughput as well compared to in vivo animal models, and compared to, it's much cheaper than animal models, and the time to result is a lot quicker as well. So one very important aspect to mention is that the FDA Modernization Act 2.0 was signed into law in December 2022, and this act amends the regulatory guidance at the FDA that requires animal testing for new drugs, adding a new section defining that non-clinical tests to include human relevant testing methods. So the MPS technology allows researchers to recreate a test environment that accurately represents the human body and generate clinical translatable data. And this provides an alternative to animals for testing drugs with specific modes of action. So to answer this need, CN Bio have created their own MPS. So this is the PhysioMimix, our MPS system. It has a unique open well design, and it's easy to install and set up. So with a controller, which is that white device on the bottom, sitting outside the incubator, and with this controller, we can set up and control the flow direction and flow rates in an experiment. So outside, we have the docking stations, which is just those metal apparatus which sit outside the incubator and contain in which we can put drivers, and in each driver sits a plate. Our MPS enables its researchers and scientists to model human biology in a lab for rapid and predictive 3D human tissue-based studies with the built-in microfluidic technology to provide nutrients and mimic blood flow. So currently, we offer we have 3 different types of plates in which we can grow different types of organs. So we have the dual organ, which we can have 2 different types of organ, including a barrier model and a liver model. The barrier plate is accessible for transvaal, so we can grow a barrier such as the lung or the gut. And finally, we have the liver plate, in which we'll be talking about today. So with this, there's different application areas such as disease modeling, safety toxicology, and anatomy studies that can be used using our MPS systems. So our liver plate specifically is an innovative system which allows us to create 3D liver microtissues for multiple applications. So the plate is designed with 12 individual enclosed recirculating wells, each of which contains a bespoke scaffold with multiple micropores. So we can seed and culture primary human hepatocytes, or PHHs, onto these scaffolds, which are constantly being perfused with a culture medium by the fluidics driven by the micropumps inside the plates. Therefore, this process allows us to provide things like nutrients, oxygen, and mechanical stimuli, which replicates the blood flow and creates a microenvironment which allows the formation of 3D liver microtissues within the micropores in the scaffolds. So this approach offers a more realistic and physiological relevant in vitro model, which has the potential to increase the translatability to human clinical outcomes. So here, just going over, we are able to show that we are able to replicate a long-term and highly functional liver model. So the graph on the right, we see that we have a production of albumin over a long period of 28 days, which stays high throughout this time, albumin being an important marker of liver health and function. And on the other hand, we have LDH with a marker for cellular damage, which is kept low throughout a long experiment. We also look at different metabolic enzymes, different CYP enzymes for liver metabolism, which are being produced at higher amounts during for different donors of liver cells. We're also able to recreate the liver microarchitecture by showing features such as tight junctions, which shows the polarity of the cells, macrovilli, as well as the lumen of bile canaliculi. We also show this to be transcriptomically stable, and like this, we are able to show also that we can replicate and establish a healthy liver model. So once we have this, we are also able to induce a different disease models such as NAFLD and NASH. So what is NAFLD and NASH? So non-alcoholic fatty liver disease, or NAFLD, is a spectrum of metabolic disorders which affect approximately 25% of our global population. Of all these people with the disease, 20% go on to develop NAFLD, NASH, which is non-alcoholic steatohepatitis. And this is the most severe form of NAFLD, which can then go on to result in cirrhosis and liver cancer if it's left untreated. So NASH is characterized by the accumulation of lipids within the liver, as well as inflammation and fibrosis. And there are currently no approved drug treatments for NASH. And the current traditional preclinical models, which are mainly based on in vivo animal models, have continuously failed to predict the human drug efficacy because they lack the human relevancy of this disease. So our NASH assay provides a preclinical in vitro model, which comprises of human primary liver cells, which are grown in 3D, and recapitulates the physical environment of NAFLD and NASH liver. So we use a triple culture of primary hepatocytes, as well as Kupffer cells, which are resident macrophages, which drive an inflammatory response, as well as human hepatic stellate cells, which drive the fibrotic response in our liver. So we culture these cells, this triple culture, within our plates, and we treat it with fat media, which contains high contents of fats and sugars. And like this, we are able to create a NASH phenotype that closely mimics the key aspects of the disease. So one of the very important aspects to talk about is our NASH experimental design. So typically a NASH experiment goes on for 14 days, being seeded at day 0, and then at day 1 we add the fat needed to induce the NASH phenotype. At day 4 we run a cell health QC, and then between day 4 and day 14 there's repeated dosing and sampling across this 2-week period, so we can do different types of anti-NASH compounds. For example, could be used to tackle and address each type of marker of interest. There's data can be generated from multiple endpoints and deliverables from our model. So we can collect at every immediate change. We can collect up to 1 ml of medium, of which we can do different types of assays, including ELISA. We can also, at the end of the experiment or during the experiment, we can remove the scaffolds and remove the tissues and do either RNA or protein analysis. And there's definitely, at the end, we do many different applications such as microscopy, histology, and metabolic studies as well. So one thing also to mention is the transcriptomic profile of our NASH model. So we have compared the profile against the human gene atlas, and we've shown that the transcriptomical profile matches the profile of human liver disease, as well as looking at close association between human NASH and MPS model. There is a better overlap compared to murine models. And as we see on the graph on the bottom, that when we compare the key gene associated with NASH, our model is a much better, is a much closer match compared to animal models. So one of the key aspects, key markers of NASH is steatosis. Steatosis being the accumulation of lipids in the liver microtissues. So here in this image, we are able to demonstrate the accumulation of fats within the microtissues. So this is a confocal image of microtissues grown inside the micropores of our scaffolds, with blue being DAPP, the cell nuclei, green being collagen type 1, and yellow being Nile red. Nile red, which binds to lipid droplets. And here we see a huge, significant accumulation of lipids within the cells in our scaffolds. We also have another image of the another image of the steatotic model, showing the thickness of the tissue grown within a micropore. So this is a confocal image of showing 8 Z stacks. So we show actually how deep the tissue is grown with it, which also sheds light to how the tissue is 3D, and we are able to recreate the NASH characteristics within our model. So we're also able to recapitulate other key markers of disease which are detectable in our NASH model. So looking at soluble biomarkers in the cell culture medium, we're able to look at different inflammatory profiles. So here we're looking at IL-6 and IL-8, which are cytokines and chemokines that are closely associated with NASH, which are being expressed at high levels due in a two-week experiment period. And we also looked at a vast range of NASH relevant inflammatory biomarkers of Illuminex, which are also being expressed at high levels during 14-day experiments. We are also able to show the enhanced production of clinically relevant fibrotic markers, such as TIMP1 and fibronectin, which similarly are being expressed over 2-week experiments. So our NASH model demonstrates the hallmarks of human NASH disease, expressing high levels of relevant biomarkers for a long period of time. This means that we can model different aspects of clinical NASH and then also investigate the mechanisms of action for new anti-NASH drugs. So here we are looking at different models. So we can also induce TGF-beta, which allows for a more fibrotic version of NASH. So basically it's an advanced NASH phenotype. And when we compare the confocal microscopy looking at extracellular matrix deposition, such as collagen type 1 and alpha smooth muscle actin, which are profibrotic markers, we see a lot more fibrotic profile of a NASH and TGF-beta model compared to just a plain NASH model. So the most important aspect of our NASH model, as we are able to demonstrate all these hallmarks, we can also look at the mechanisms of anti-NASH compounds. So here we are looking at elafibranor, which is a drug that was in phase 3 clinical trials when it was dropped, and we are able to see a significant decrease of reduction in inflammation, fibrosis, and steatosis, so all of NASH hallmarks of disease, which were observed following treatment with elafibranor. So we are also able to look at the different dose-responsive effects of both elafibranor and obeticholic acid, which is another drug that failed at those at previous stages of clinical trials. But we are able here to see that the responses at previous stages of clinical trials, the responses are the same as we observed, as we have seen here. So as a dose response, we see that the NASH model recapitulates the key aspects of NASH disease, and the dose response also matches with what we see also in the results within the NASH model. So therefore, as we are able to recapitulate the key aspects of NASH and use it to predict human response to anti-NASH therapeutics, we're also looking at other anti-NASH compounds which are currently being screened. So in summary, we have shown that our NASH model is a closed model that replicates closed human NASH pathophysiology. So it contains the correct, the appropriate cell types appropriate to NASH, and it is being induced by fat medium, and it is grown in a physiological relevant microenvironment with fluidics and the stimuli required to induce this disease. With this model, we are able to have a more accurate prediction of the human response to drugs compared to animal models, because it contains human cells and we can predict the responses in much better because of the better mechanisms that human cells have that animals don't, and therefore providing a better in vitro to in vivo translation using the clinical relevant biomarkers such as fibrotic and inflammatory biomarkers discussed in this presentation. And finally, it is highly adaptable to specific aspects of disease, being able to demonstrate a NASH model as well as an advanced NASH model with TGF-beta, which induces a advanced fibrotic phenotype. And that's all from me for today. Thanks very much for listening to my talk today. I'd like to thank my colleagues at CN Bio, and I'm happy to answer any questions. You can contact us via email or visit our website. Thank you.